Hello students, welcome to EPG Pathsala of University Grant Commission of India. I am Dr. Sohail Akhtar from Assistant Professor from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Ahmedabad, New Delhi. Today we are going to discuss about a module continuous release system under the paper Novel Drug Delivery System 1. So students, let's see what we are going to study in this particular module. In the continuous release system, this is a kind of controlled drug delivery system for oral delivery. We are going to discuss dissolution control type, diffusion control type, combination of dissolution and diffusion, ion exchange resin drug complex, slow dissolving salt and complex, pH dependent control release formulation, osmotic pressure controlled control release formulation, and last but not the least, hydrodynamic pressure control delivery system. In the second category, we will discuss the controlled drug delivery system based on delayed transit, and these can be done with the help of altering the density. So we call this altered density system. That can also be achieved by mucoadhesion with the GIT tract, and we call this mucoadhesive system and the size based system. So let's start with the continuous release system based on dissolution, and accordingly it is called as dissolution control system. The drug dissolution rate is controlled either by the process of microencapsulation or by the use of slow dissolving or solubilizing polymer. Once the coating material is dissolved in this system, the drug become readily available or drug become available for the process of dissolution. So by varying the thickness of the coat and the composition, the drug release can be modulated and toned up. Some preparations contain a fraction of the total dose as an immediate release components to provide a pulse dose after the administration. The pellet dose form of the diffusion or dissolution control product can be encapsulated or prepared in the form of tablet. It is indicating about dissolution type control release system. So basically what we have studied and what we are studying is all about the control release system. So control release system based on the route of delivery, it could be oral route, transdermal route, injectable route. So there are different route. On that basis you can categorize this control delivery system. What we are going to discuss here now is an example of oral route or oral control release system. It could be on the basis of release controlled mechanism. It could be dissolution type, diffusion type, combination of diffusion and dissolution type, and there are others as well that we will learn or will study in the later slides. Here in this slide is an example of dissolution type control release system. In this case, mass transfer from solid to liquid, and rate limiting determining step is a diffusion of solid toward the liquid. So how the design of such dissolution type control release systems is of two kind, majorly a matrix type and another one may be an encapsulation type. So what is in the matrix type is the topmost layer is dissolving coat, and then layer by layer drug layer actually is in the layer by layer, and each layer is separated from other layer with the help of a dissolving coat again. In the case of encapsulation type, the drug is dissolved state encapsulation within the core, and outer layer is comprises of dissolving coat, which is relatively, and it depends, I mean, if you want to have a what kind, depending on your control release pattern and what kind of control you want, to what extent the thickness of this diffusing coating layer is changed. It can be thinner, it can be thicker, it can be more thick. So in this slide we are going to discuss these two type of dissolution control release system. The one encapsulation one is drug is encapsulated in a slow dissolving polymer. Next one, in this case, so the rate of dissolution depend on thickness of the microencapsulation coat and hydrophobic additives which is used in its preparation. The other one, which is the matrix type, drug is uniformly dispersed in slow dissolving polymer. So basically in matrix 1 drug is dispersed, so drug is not in dissolved state. Okay, it's in encapsulation type, drug is basically dissolved. Okay, so this is the basic difference between these two types. So in the matrix type, the release, how the release govern and how the release depend on some important factors. Important factor of drug release depend on porous nature of polymer, penetration rate of dissolving material or dissolution material, and the composition and wettability of the surface layer. Previously, you learned about dissolution control. In the dissolution control, you learned that it can be encapsulated one, it can be the matrix one, and how the drug release from there. The another type of controlled oral controlled release system is based on diffusion. So it called as diffusion type oral control release system. At the name itself indicate that drug release is governed by diffusion process. So drug molecule diffuse from the region of higher concentration to the lower concentration until and unless equilibrium is maintained. This is the basic mechanism behind the controlled release pattern from such system. Therefore, in this case, the release is directly proportional to the concentration gradient across the membrane. When the concentration gradient is finished or when the equilibrium achieved, diffusion of drug or release of drug is stopped. It again, just like dissolution control, it again of two type, reservoir type and matrix type. Similarly, just like the previous one, reservoir, in the reservoir one, drug is dissolved state in the core, and in the matrix one, drug is dispersed in the core. Okay, so this is the same system what we have learned before. So drug, the first figure here is indicating drug is in the core and the ghost matrix. So the drug, there is a layer from where the solution goes in and has started dissolving the solid material. So there is a high concentration zone of the drug and there is a zone where it is almost no concentration of drug. So, and there is a middle matrix zone which is having some, until and unless equilibrium is not maintained, this is the diffusion layer for the drug. Now the second one is diffusion type control release system. As word itself indicate that it is actually the diffusion which governs the basic mechanism behind the release or controlling of release from the dosage form. In this system, there is a water insoluble polymer which control the flow of surrounding fluid or biological fluid and the subsequent release of dissolved or dispersed drug from the dosage form. The process of diffusion starts when the drug passes through the polymer that from the control release device. The process of diffusion can occur either through the pore present in the polymer matrix or by passing between the polymer chain. These are broadly divided into two categories: reservoir type and matrix type. In the case of reservoir type, drug is encapsulated within water insoluble polymer. Now, drug partition into the polymer membrane and exchange with dissolution fluid, and finally the drug release rate remain constant in particular reservoir type system. So this system can give you the zero order release kinetic pattern. Now another one is the matrix device. Drug is dissolved, drug is dispersed within the polymeric matrix, so it is not in the dissolved state. Drug is in the form of suspension. Diffusion occurs when the drug passes from matrix to external environment. So in this case, release rate decrease over the period of time. As I told you before, it can be, the system can be dissolution and diffusion, or it can be the mixture of both. So this is the example of oral controlled drug delivery system which having a combination of mechanism of dissolution and diffusion. So it is called as dissolution and diffusion type controlled delivery system. How it look like? The structure showed here. It contains the core. Core is either dispersed drug or dissolved drug within the polymeric matrix. Topmost layer or outer layer, which is the surrounding layer, is comprises of polymers or material which can be semi-permeable and mixed with the material which can be dissolved also. So once it is in contact with the fluid or biological fluid, the material that can be dissolved, it form the pore. Okay, So the pore is created by dissolution fraction of soluble membrane. Remain part is semi-impermeable membrane which is not going to be dissolved, and this pore actually help in coming out or releasing of the drug. So and this pore also help in the entry of dissolution fluid, and then drug diffuse through the same pore by the process of diffusion. So this is the explanation of the previous slide what we have seen in the figure. Here it is indicating what is happening in such system. So in this system, drug is encapsulated in the partially soluble membrane. Pore are formed due to the dissolution of polymer on the surface. Now dissolution medium or biological fluid going into the core, dissolving the drug, and this drug is coming out with the help of diffusion process. So here some of the example of polymer used in this such kind of design are ethyl cellulose and polyvinyl pyrrolidone mixture. Abbreviation is PVP. Earlier you have seen that as an oral controlled drug delivery system, you have seen dissolution, diffusion, and combination of both. This is another example of oral controlled drug delivery system called as ion exchange resin. Itself it indicates that there is some process of ion exchange. It means that there is some ionic active or ionized material attached with the system that help in exchanging within the biological fluid that contain different kind of ion. So here it mostly used for the drug which are ionized or ionic in nature. You can see here in brief I'm discussing with you that this system is drug resin complex. Basically this is drug resin complex. It is known as resonate. So resin are basically water insoluble cross-linked polymer, and it have a salt forming functional group representing position on the polymer chain. This complex, so this form the complex with the drug actually, and this complex is not a ionic bond, but basically it is something with which the other ions from the biological fluid can exchange with the drug, like sodium chloride, calcium, whatever, it depend on the situation. These complexes are prepared by repeated exposure of the resin to the drug in a chromatographic column, or by prolonged keeping this resin in contact of drug solution for prolonged period of time. Indicate that how the formation of drug complex happening in such system or ion resin system. As I already told you that this polymer is ionized in nature or carrying some charge. So drug can interact with the charge depending on the nature. If the resin is negative, positive drug can interact with it. If the resin is positive, negative drug can interact with it. And then further the drug exchange happening how? Drug exchange happening with the ion within the biological fluid. So if the biological fluid contain chloride ion is getting negative charge, it can displace a negative charge drug and bind to the positive charge resin polymers. So the diffusion process in the reaction we explain here. If the resin is positive, drug must be negative. It is well understood, okay? And then negative charge component from the biological fluid displace the drug, negative charge will displace the negative charge molecule and bind with the resin. So negative charge drug is released by this way, and vice versa in the case of positive charge drug. This is an example diagrammatic presentation what we have discussed. You can see that a drug in hydrochloride form bind with the resin, okay, that is resin or drug complex. During the release, drug hydrogen ion from the biological fluid bind with the drug and NaCl, and then finally drug is released with the HCl, and Na from the NaCl goes to the resin. So in this case, what are the different kind of ion exchange resin, which is the main and most important component of this regular system? Are ion exchange resin can be, as you know, that it must be charged, okay? So charge, there is no different kind of charge, only either positive or negative. So positive charge is called cationic and negative charge is called anionic. This cationic charge containing ion exchange resins are either from inorganic category or either from organic category. In organic category, cationic resins are either natural or synthetic. Natural example of natural one is clay, and synthetic one is zeolite. Or from the organic category, maybe it is again natural, synthetic, or semi-synthetic. Example of natural cationic organic ion exchange resin is lignite. Semi-synthetic one, the most common is zeocab and cephalodextrin, and the synthetic one is acrylic acid copolymer or acrylic acid derivatives. Now come to the anionic one. Again, just like cationic one, it is again coming from inorganic category or organic category. From the inorganic category, it might be natural or synthetic, and organic as well, semi-synthetic and synthetic. So there is no natural, organic, anionic ion exchange resin. Okay, keep in mind. So from the inorganic anionic exchange resin, example from the natural one is dolomite, and synthetic one is heavy metal silicates. From the organic category, semi-synthetic one is Sephardex, and from the synthetic category, it is amino formaldehyde resin. The next example of oral controlled drug delivery system is pH dependent system. So in this case, weakly acidic or basic drug shows pH dependent solubility at different pH of gastrointestinal tract. This is well known fact. Drug dissolution rate is in this case is proportional to the solubility of drug in that medium, and pH dependent solubility resulted in different dissolution rate in different part of the GIT tract. Buffer such as the salt of amino acids, citric acid, phthalic acids, phosphoric acid and tetratic acid can be added to the formulation to maintain a constant pH. So these are actually the buffers that are using, thereby rendering the pH dependency drug release. These kind of systems are prepared by mixing a basic or a acidic drug with appropriate pharmaceutical excipients, must be inert and biocompatible obviously, and coating further it with coating with the GIT fluid permeable film forming polymers. So far you learned about different kind of oral controlled drug delivery system that are dissolution control, diffusion control, combination of both, ion exchange based. Now we are going to discuss another one. It is slow dissolving salt or its complexes. Itself name indicate that drug or chemical drug or substance of interest is converted into the form of a salt which is slow dissolving. It means its solubility in the water is less compared to the drug itself. So this is the technique normally used to make controlled delivery or controlled release pattern for a highly water soluble drug. So such complex can be obtained by simple acid base reaction or maybe by some complex chemical reaction. Okay, some example of such system available in the market are oral suspension of benzathine penicillin G, which is basically the chemical complex of penicillin G with NN-dibenzyl ethylene diamine. Now this is another separate section or separate example of oral controlled drug delivery system, and it is osmotic pressure control. We have already discussed in separate model about this system in a well elaborative and extensive manner. So I will just summarize you here in a brief way. It is also called oral osmotic pump or OROS. So it basically work on the principle of osmotic pressure. How it happened? It definitely contain osmogen or osmotically active material in the core mixed with the drug solution or in a separate layer. Once it is coming in it is exposed to the biological fluid or GIT fluid, it dissolves the osmotic active material or osmogen, and this finally increase the osmotic pressure that help or push the drug to come out or dissolved drug come out from the orifice. So this is the basic diagram that indicating the assembly of osmotic pressure control system. A typical diagram of osmotic pressure control system. There's two kind of. One is in which osmotic core contain drug and osmogen there is, and separated or covered by highly semi-permeable membrane, and then there is the orifice. So semi-permeable membrane allowing water to go in, or biological fluid or aqueous fluid to go in, and it dissolves the osmogen, pressure create, and it expand. By that way it want to come out. So solubilized drug within the water is coming out through the pore. In the second diagram you can see that semi-permeable membrane having a drug solution in the core, osmogen is in a separate layer. So water first coming in contact with the, from the semi-permeable membrane, it is coming in, dissolving the osmogen, and after dissolving the osmogen, it increase the intra-osmotic pressure. Now this osmogen layer and drug layer is again separated by semi-permeable membrane. So it indicates you that water is allowed to go from the osmogen layer to the drug layer. So once the drug is there, drug is started dissolving or solubilizing. With this pressure from the osmotic zone, it pushes the drug out of the orifice, and this is how it releases. The equation that governs this release pattern is given here. So mathematically we derived and stated it. Rate of flow of water equal to Ka upon H delta pi minus delta P. So what is this? This is the rate of flow of water, which is directly proportional to the release of drug from the pore. Okay, so rate of drug release. So basically it is helping in calculating the rate of drug release. Here K is the permeability coefficient, A is the effective area or surface area, H is the thickness of semi-permeable membrane, delta pi is osmotic pressure difference, and delta P is hydraulic pressure difference. It indicates here that rate of water flow is directly proportional to the effective area, and it is inversely proportional to the thickness of semi-permeable membrane, and directly proportional to the pressure difference. As much as the pressure difference is, rate of release is more. If thickness is high, rate of release is less. This is another example of oral control drug delivery system. It's more or less similar to the osmotic pressure control. However, here the pressure control is not governed by osmogen but by hydrodynamic pressure mainly. So what is hydrodynamic pressure? It is the pressure created by excess of water come in and create a pressure difference. So basically this is made by using a material, a polymers that can have the water soluble property. So basically hydrophilic, so as I told you, water soluble property, here it is a best example. Hydrophilic gums are normally used in this case. Once they get swell or wet, they create pressure, or because of change in the shape or because of their expansion, it creates pressure. In the core or in the system, there is a drug which is dissolved in biological fluid or water. Similar to ortho system, oros system, there is an orifice and it helps to push the drug out. This is a simple structural diagram indicating the working and structural assembly of hydrodynamic pressure control system. The another example under the category of oral control drug delivery system is hydrodynamic pressure control system. It is a collapsible impermeable container containing liquid drug solution that form a reservoir compartment which is covered inside a rigid shape retaining housing. In such system, composite laminate of a absorbent layer and soluble hydrophilic polymers layer is sandwiched between the drug reservoir compartments and the housing. At the site of absorption, the laminate absorbs the medium through the annular opening at the lower end of the housing and become increasingly swollen, which generate hydrodynamic pressure in the system and leading to the release of drug from the orifice. So this pressure ultimately forces the drug to release through the delivery orifice at the specific rate. This is also called as or known as push-pull osmotic pump. Another category of oral control drug delivery system is delayed transit and continuous release system. Word itself indicate delayed transit. So it is delaying the transit. Transit means passing through some area. Okay, that is delayed. How you can make such a system? So basically it is increasing the transit time from the GIT tract. How it can be developed? It can, based on their way of developing, they are of three types. First, changing in the density, altered density, high density approach, low density approach. Second one is mucoadhesive system, and third one is the size based system. I will explain you later how it works. In the altered density system, high density approach is in which we use some heavy metals which are relatively higher weight. Because of that high density, it settled down and stuck into the stomach region and remain in that for longer time. Low density approach is we are using the polymers that can absorb the water and swell and start floating. So when this start floating on the gastric fluid, it takes time to pass. Okay, so this is how altered density work. In mucoadhesive system, we use mucoadhesive polymers in the design of such drug delivery system. This mucoadhesive, mostly they are positive charge or bioadhesive itself. They bind with the mucus or mucin of a stomach or GIT tract and stuck there and remain there. So it is basically two-way, either electrostatic interaction and bioadhesion with the mucin layer or mucus layer. Size-based, they are relatively bigger in size, okay, that slowing passing through the channel or tubes or the pathway of the GIT tract. So students, okay, so let us summarize this particular module, what we had studied so far. We studied here about the oral controlled drug delivery system, and that is based on diffusion, dissolution, or combination of diffusion and dissolution. These are all the mechanism actually. On the basis of this mechanism, we named it or we categorize it. Other example is pH control and ion exchange complex, slow dissolving salt. Not only this, we can also develop such oral controlled drug delivery system by modulating the size, by modulating or improving the mucoadhesion within the GIT tract, and also by altering the density, by increasing the density of dosage form or carrier system, or putting high density material inside, or floating system, or let's say low density, which is an example of floating system. We had already discussed in such system we use polymer which swell and start floating, and by this way it remain within the gastric fluid and delaying the transit through the GIT tract. Thank you so much.